Brown developed a technique where cDNAs can be embedded onto glass slides. Using this DNA arrays, he could do large-scale expression studies. Growth stage specific mRNAs are isolated and then reverse transcribed to give unique cDNA populations. These are directly embedded onto specifically coated glass slides. The slides are coated with polylysine, which is positively charged. DNA is negatively charged, so the cDNA sticks to the slide through an ionic interaction. The cDNA can still interact with the DNA probe. Brown used arrays to obtain gene expression profiles of cancer. Diffuse large B-cell lymphoma is a common lymphoma cancer of lymph nodes. He found subtypes of DLBCL that correlated with the survival rates. Brown made the chip with genes expressed by lymph nodes and those important in cancer biology. A total of 17,856 cDNA genes are printed onto what we call as a lymphochip. Remember, each square of the chip corresponds to different cDNA. Then he made a cDNAs from different DLBCL tumors. He labeled one set of cDNA with red fluorescent tag, the other with the green tag. Brown incubated the arrays with the tagged cDNAs, which bound to the matching genes printed on the array. Since he knew the position of the genes on the DNA array, Brown could figure out the level of gene expression based on the color signal. If the gene was expressed in DLBCL1 cells, the square was red. Similarly, if the gene was expressed in DLBCL2 cells, the square was green. If the gene was expressed equally in both cells, the square was yellow. Thus Brown identified two subtypes of DLBCL, GCB-like and activated B-like DLBCL. These subtypes have different response to therapy, and with this type of diagnosis, more tailored treatment can begin for patients. This type of tailored treatment is called pharmacogenomics. Brown can now also analyze the difference in gene expression of these two very similar lymphomas. This may give a better understanding of how cancer works and hopefully develop a better therapies and cures.